So I'd like to tell you two stories. The first is actually going to be a technique-driven story. It's about a technology that my lab has been developing to address the kinds of questions that we've been after for a very long time, but we didn't think the tools were out there. And the second will be bread and butter visual cortex computation. So hopefully you're also interested in that. Okay, so what I realized is when I started my position at Berkeley is that the tools that many of us have been and are still using to manipulate neural circuits were basically inadequate to address the kinds of questions that we really wanted to address. In particular, my lab is interested in the neural basis of sensory perception. And I'm going to tell you about a project that we worked on to be able to control neural activity with extremely high spatial and temporal precision. So these are the people that actually have done the work. Actually, Nico Pagard does the first. He's an optical physicist. He just on the job market has a number of offers, and he's co-advised by my collaborator Laura Waller, who's in engineering. So they actually did the heavy optical physics side of this project. And then Allen and Ian are neurobiologists trained at Harvard who actually did the experimental work, and Savita did the ops and engineering I'll tell you about. Okay, so the broad question is, how do we crack a neural code for sensory perception? And so the question is, how does the brain generate a sensory perception? And what neuroscientists have done for decades is to correlate the activity of neurons in the brain with a specific sensory stimulus. But this is purely correlational. You can't derive causal relationships between patterns of activity in the brain and a specific sensory percept. And so for this, we wanted a new technology to actually achieve this. So I'm sure you're familiar with the various approaches we have, like optogenetics, to manipulate activity. But they didn't have the precision to actually relate precise patterns or precise features of activity to a specific sensory percept. And by developing this technology, we could not only address this question, we thought it would be broadly useful in neuroscience and potentially as an optical neuroprosthetic. Okay, so basic question from a neurobiological perspective is if you have an image like this where you can have two different and competing visual percepts, the idea from neuroscientist perspective is that you have a bunch of neurons schematized here, say in a higher visual area, that's going to fire in a certain way when you see the faces, but even given the same retinal image, you get a different percept when you see the cup. You have a different pattern of activity of those cells or of different groups of cells in the same cortical area. While we can record these activities with electrodes or imaging, how do we actually prove that these features, what features of these patterns are actually driving sensory percepts? And so we need a system that can both read and write the neural code simultaneously in the brain while an animal is actually perceiving different stimuli. So reading the neural code, pretty straightforward. This is just calcium imaging with GCAM. That's the tool we'll use currently. It has some inadequacies, which I won't really address. Working on that. But mostly, this is about how to write the neural code. And so the dream, of course, for writing the neural code is to have the same spatial and temporal resolution at which neural circuits operate, which is single cells, single spikes. So most of you are probably familiar with optogenetics, expressing microbial opsins in neurons to make them light sensitive. This is a rendition, artist rendition of optogenetics from Ed Boyden's lab on his website. But this, of course, is fantastical because this is not what 99% of optogenetics looks like. It looks more like this, where you put a fiber and your specificity doesn't come from where your light is shined, but actually comes from the genetic specificity of, say, your driver. So very powerful, but in the end, what most studies, or nearly all studies, have done is correlate a specific cell type's activity with a specific behavior or percept or so forth. What we needed is actually something to achieve at this level of precision, so single neurons, like a spotlight on the brain. And so what do we need to achieve this? So the first and most important is stimulation on the order of cellular resolution to differentiate individual neurons in large ensembles. We need to have millisecond precision. And so it's a major debate in the field of neural coding how much spike timing precision matters in the cortex, but we need this ability to control spikes with that level of precision if we are even going to ask that question. We need to operate in 3 dimensions. This seems obvious. It's because the cortex in any circuit is inherently 3-dimensional, but most optical imaging systems sort of have a geometric focus that's 2-dimensional. And finally, it has to be compatible for functional imaging like calcium imaging. So given these constraints, what we can turn to is multiphoton optogenetics, which is where you're going to illuminate the microbial opsins with 2-photon light, taking advantages of its axial resolution and its penetration through brain tissue. So very briefly, to summarize a bunch of work by other labs in the field, some of the first work was simply taking your scanning microscope with a laser beam, that's a tiny little spot, scanning on a neuron soma and driving enough current over time to spike the cell. The problem here is that it takes many milliseconds to spike a neuron, so you don't have much temporal resolution. So you can turn to a technology called holography, or digital holography, pioneered by a number of groups like Valentina Miliani, in which instead of scanning a beam, in an instantaneous flash of your laser, you shine photons all over the cells of interest. The problem with this technology is that while in principle it gives you temporal precision, it actually has fairly poor axial resolution despite the fact that it's two-photon technology. So Emiliani and a few others came up with a solution, or employed a solution called temporal focusing, which I won't get into for the sake of time. But what this does is it takes the same pattern of light and restricts the excitation to a single plane. But the problem is, while you improve your axial resolution, you lose your ability to work in 3 dimensions, which is the major benefit of holography. So we came up with a technology I'll just briefly get into, which we call 3-dimensional scanless holographic optogenetics with temporal focusing, or 3D shot for short, which allows us to regain this ability to shine very precise spots of light in 3 dimensions. So the basic principle of holography is you take your Gaussian wavefront, your laser beam, and you diffract it off a grating for temporal focusing. But the key point here is a device called a spatial light modulator, which is a bit like a glorified projector that works in phase modulation. And because it works in phase modulation, you have the ability to focus light at different points in space in 3-dimensional volumes to target specific neurons in the cortex. So given the constraints, we had to come up with an alternative system to actually achieve this that would still give us the proper spatial resolution. And so Nico in the lab paired this is a conventional 2-photon microscope, with this slightly altered holographic stimulation system that I won't fully explain. But suffice to say, I'll show you what it actually achieves. And so here is a rendering on a thin fluorescent film of a 3-dimensional hologram. This is literally a pattern of light that's generated in a single shot of your laser that you can project directly into the cortex, where the position, number, density, and so forth of these spots is entirely determined by the user. You simply say, you pick your cells in 3 dimensions, the cells that you want to stimulate, and it generates a copy of an image about a disk of light that's targeted approximately to the size of a neuron, and you could put it anywhere in a relatively large 3-dimensional volume, currently spanning around 500 to 500 by 500 microns. So this shows you different numbers of spots. You have a lot of control here. The key is that these are still fairly resolved in the axial dimension. You can take the same number of spots and simply change the density of those spots. So here you're at a density of basically hitting every neuron in a little volumetric patch of, say, a cortical column. Or you can span that out over several columns and so forth. Yeah. Yes, exactly. That is true. So what happens is you go up to 500 to 600 spots, your contrast ratio for the light in your target and out of your target gets worse. And so I'll get to this later, we only do about 50 spots at a time. But that's the key idea. This is per frame of the spatial light modulator. You can update the spatial light modulator now at around 200 to 300 hertz. So you can temporally multiplex your stimulation to overcome that challenge. The total power that we use through the objective and average power is about 4 watts into the brain, but it's duty cycled so that you end up with about a sixth of that. So under a watt. Okay, so on the order of a scanning microscope. So we can also, I guess one key feature of this is that it works pretty well through scattering. And that's the key principle of 2 photons. So this is the exact same hologram projected through different thicknesses of brain tissue. And you can see that you lose a lot of energy. But what you don't lose is resolution. And that's important. So even down to about layer 4 of mouse, 400 microns, you basically preserve the overall shape of your point spread functions. That's the sort of profile of these spots. You need to crank up the laser power, which is then scattered. So of course, that is a constraint, how much energy you can put in the brain. Okay, just to show you from making electrophysiological measurements, you put a loose patch electrode on a neuron that's expressed in the opsin under 2-photon guidance, and we compare conventional holography, which is in black, to temporally focused 3D shot, which is in red. So the latter resolution doesn't get any better, that's expected, but the axial resolution gets dramatically better, such that it's compatible on the order of stimulating individual neurons, like one, but not the one immediately below or above. Okay, so with this technology, we have basically scale on the order of single cells. We can operate in 3D. But actually, it turned out to be a major challenge was to do this with millisecond precision and to do this at many neurons at the time. And the major constraint actually was the actual microbial opsin, the protein itself that absorbs photons. And so if we want a system that can achieve our desired goals, we need an opsin protein that is extremely fast, fast on, fast off, extremely strong, and also has the right spectral sensitivity so it can be compatible with, say, GCamp6 imaging. So we looked at a bunch of opsins. We probably, before we did any work on this, we probably scanned maybe 10 to 20 different opsins that we'd found in the literature. This is a figure from a paper from Ed Boyden's lab where he cloned a whole bunch of opsins from different algae species. And we said, well, the opsin that we want should be very fast and reasonably strong. And it turns out that Chronos, for those familiar, is the fastest opsin naturally occurring that we are aware of. It's extremely, extremely fast, faster than cheetah or anything like that. It's a little red-shifted channeled opsin, so its 2-photon spectrum is nice. It's about 1,000 to 1,020 nanometers as its peak absorpance. And it's reasonably strong. No, not really necessarily much stronger than channeled opsin, but still pretty good. So we said, OK, let's take this opsin and see if it's good enough for our purposes, comparing it to several other opsins. I'm going to make a very quick aside if Chris Baker's here. No? There he is. So Chris gave us a soma-targeted opsin that he had made a channeled opsin. This is actually our own version of Chronos fused to a red protein. This is a wild-type Chronos, and this is the KV2.1 sequence. And this is a total aside that Chris and others had showed that by basically with this sequence tag, you can aggregate or congregate the opsin basically on the soma and minimal proximal dendrites, which is a huge, huge advantage, not just for the spatial resolution of one cell, but even more importantly, if you want to stimulate 50 cells, if you have opsin on the dendrites, you're pretty much screwed. You actually have to do this. There's no choice. So this actually works. It also gives you more photocurrent when you're targeting the light to the soma. OK, so every opsin you'll see in the rest of this talk is soma targeted. OK, so it turns out the existing opsins, and we chose some of the most popular, C1V1, Crimson, Chronos, all soma targeted, gave you reasonable photocurrents when you illuminated them with light. This is a patch clamp recording of the photocurrent. But the problem was to spike a layer 2-3 cell in the cortex, an unfortunately fairly inexcitable neuron, you need, with a short pulse, about a nanoamp of current. That's a lot of current, and you just weren't getting that with these opsins. Now, if you turn on the light for a long time on one cell, you could maybe get them to fire, but still, a third, 2/3 of the neurons weren't even spiking under any conditions until you vaporize them as you turn your laser higher, which wasn't good. So reluctantly, I realized that there was no choice. We had to make better opsins. And Savita, in a little bit of collaboration with a colleague of mine, Steve Braun, who's a structural biologist for Rob McKinnon's lab, we began to look at the secondary sequence of opsins and look at models of the 3D structure. So Savita lined up all the secondary sequences of a bunch of cation opsins we had worked on. And she noticed in the structural model that Steve helped us build is that there was a very interesting residue that in every cation opsin except for Chronos was a glutamate. Like channeled opsin, Crimson, they both have glutamate. C1V1 has a glutamate. But Chronos, in this residue that putatively lines the conduction pore and interacts potentially with this retinal chromophore, is a methionine, very different amino acid. So that really sparked our interest. It turns out for the aficionados here that residue was mutated in cheetah to make channeled opsin faster but much weaker. So we mutated this residue, methionine to glutamate, called it chrome. And the bottom line is that a single point mutation gave us almost 4 times more current. Now, it slowed the opsin down a little bit, about 20%, but it made the current 4 times larger. I won't speculate fully about why we think this might be, but it seemed like Chronos was optimized for speed, and to compensate for the optimization of kinetics, other residues had to change to make it strong, so we can go back to the glutamate to make it slightly slower, but get a huge current. Anyway, in practice, this works great. You get for most pyramidal cells, you get 1 to 2 nanoamps of current, which is well above what you need for 5 milliseconds to spike the cell, and basically now you can spike both in slices and in vivo almost every cell with a reasonable amount of energy well below the toxicity threshold. So that was key to using this opsin. I think we've given this to Honghui and making a reporter for this. So it seems to be the fastest, strongest opsin that we are aware of. So, it's a little bit indeterminate. They both traffic to the membrane well. We can't measure single channel conductances, so it's difficult to answer that question. Because it's in the pore interacting with the chromophore, more likely it's something to do with conduction. But you're right. It could be due to trafficking. We don't care, I guess, as long as it works. Just to look at the kinetics, these are peak scale. We're just looking at the shape. C1V1 is a very slow opsin, so it's good for scanning, like Rafa Eustace and a few other groups have used, but not so good for holography. Crimson is a nice opsin. It's faster, but still pretty slow, whereas Chronos is already desensitizing before these things have even peaked, so super fast opsin. And Chrome is slightly slower, but still extremely fast. The key thing is if you give a holographic pulse and spike a neuron, when you can, C1v1 cells fire somewhat willy-nilly, chromatin a little bit better, but if you look at Chronos and Chrome, you get very, very precise spiking. To quantify that, here we'll just look at the jitter. The jitter is the standard deviation of the spike times relative to the light pulse. You see that for these slow opsons, when you spike the cell, it's kind of a little bit highly variable, but Chronos and even Chrome, which is even stronger, and that's probably why it's faster, you can get sub-millisecond jitter, maybe sometimes in the order of 200 microseconds. So really, really, really precise spike timing. So the question we wanted to ask is, if we have this control in vivo, can we replay a very specific pattern of spikes to the brain to mimic a physiological spike train? So neurons in the cortex, as many of you probably know, fire in a somewhat Poisson-like statistics way, where they're somewhat random when the spikes occur. So we generated random spike trains. This is a raster plot, trials by time, of light pulses. We then make a 2-photon targeted patch from a neuron expressing Chrome. And these are the spikes we get out for each of these light pulses. This is the overlay. And if you quantify that, about 96% of the time that you ask for one spike, you get exactly one spike, not 0, not 2. And the jitter, the deviation of that spike time from the light pulse, is about less than a millisecond. And so this obviously gives us the ability to recreate patterns of activity in the brain that are even much more reproducible than the brain would ever exhibit, or at least the cortex. Okay. Just to extend that, if you look at GABAergic neurons, which fire at much higher rates than pyramidal cells, for this we're actually using Chronos, and I explain why Chronos later, but it turns out interneurons are pretty easy to spike, so you don't need those huge currents. With our 3D shot approach, whether it's SOM via PPV representing the majority of interneurons in layer 2-3, you can get very reproducible precise responses such that the jitter is under a millisecond, the spike probability is very high, and here you can look at spike probability as a function of instantaneous stimulation rate, that's the instantaneous frequency, and at least for PV and SOM, you're reliably generating spike trains up to 60 or more hertz, which is roughly how these cells fire to sensory stimulation in vivo. So you can reproduce the patterns of activity that these cells would normally exhibit. Okay, brief aside, while I've been talking mostly about generating spikes, which is going to give us this gain-of-function approach, you'd want a loss-of-function approach as well to remove spikes during, say, a sensory perception and see if that neuron was important. So to do this, we looked at inhibitory opsins, things like halorhodopsin or archaeodopsin, and they're pretty bad. So you get tiny currents. These are pumps. These are not ion channels, and we really couldn't do much with these. Once they're soma-targeted, that is. If they're not soma-targeted, you actually can get enough current. But as I mentioned, that's really important for resolution. We then looked at channels. So this is a naturally occurring red-shifted chloride channel. This is one engineered by Deiseroth pretty amazingly from channelrhodopsins. So it's fluxing chloride. But even so, those currents were weak. But if we looked at the naturally occurring one called GTACR1, cloned by the Sputnik lab, you get these massive currents in neurons. It's almost several log units larger than the inhibitory pump opsins that most of us use. So that was really crucial. And importantly, as a channel, it's going to deliver a shunt and not just a hyperpolarization. So here, we're making loose patch recordings from neurons expressing these opsins. And with this soma-targeted GTACR, you can basically eliminate spiking entirely. Huge currents when you make whole cell recordings from these cells. But there was a major problem. And the problem we actually kind of anticipated anyway is that when we started expressing these GTACR proteins, the cells would just die. So unlike the cation opsins, for whatever reason, the protein aggregates intracellularly, unlike channelrhodopsin, which kind of goes to the membrane nicely. So there was a huge trafficking problem. We thought maybe that's why the cells would disappear. So long story short, I'm not going to tell you about all the ins and outs, but we decided to lower expression using an IRES sequence, as well as add a bunch of trafficking motifs that had been done for pumps. And here we're just looking at the nucleus, which is labeled red. And for months, we can now see that these cells survive. They still have almost the same size currents, despite lowering, in theory, the overall expression. And the important part is that for relatively low energies, even lower than excitation, you can get potent suppression of spikes in vivo. So here's a raster plot. The laser comes on. The cell stops spiking. It's got pretty nice temporal precision when the cell stops spiking, and you can actually get prolonged suppression even for short pulses, which could look as a benefit or a pro or con, depending on how you look at it. But the bottom line is you can effectively suppress these cells even with relatively short pulses. So a very useful technique for eliminating spikes with the same spatial precision that we could drive them. I'll skip that. Okay. So back to the question at hand, which is we want to generate a system that can do what we call all optical read-write, simultaneously measuring activity and then putting that activity pattern back in the brain. So you build a scope with an imaging system as well as a holographic system that are then combined. They're two different color lasers, one at 930, one at 1040. And in this case, what Allen was doing is he was just picking a bunch of cells. He's now doing everything in 3 dimensions, both with scanning and with holography. And for each neuron, he can sort of build up a dose-response curve, how much energy you need to put in that target. And of course, each target, by the way, can be independently modulated in your hologram. It's not just binary. It's actually grayscale. And you can say, I want to pick just the right amount of energy to spike that cell 90% or 100% of the time. And so this is a movie of what that looks like for one cell at a time, where the arrow indicates where we're going to stimulate, and the red is the delta F over F of these GCamp expressing neurons. So the neurons co-express GCamp and Chrome. So they can be imaged and photostimulated. And we're doing it simultaneously in 2 planes. And this is one cell at a time. So you can look at the calcium raster plot of these cells, cell by cell by cell. You can build up a sequence. Allen built up a sequence, in this case, of 134 neurons. These are individual trials where red indicates firing and the diagonal is the cell that he's targeting. So by and large, you're getting very reliable, reproducible responses to the cell that you're interested in. You do get some other effects, maybe a little bit off target, but a lot of that is probably synaptic effects. Okay. In fact, if he goes around and actually stimulates individual neurons with different numbers of action potentials to ask this sort of like elementary question, what's the impact of one neuron on the network? And he did it with different numbers of pulses at a defined frequency. What he found is, okay, here's the neuron that actually is targeted. It responds with a huge change in calcium. But there are a bunch of neurons that are quite far away. They could be 100 microns away that will show a change in their evoked activity. So it turns out that actually this experiment had been done by my colleague Yang Dan, but with patch clamps. She patched a neuron, drove different numbers of action, well, her postdoc patched the neuron, and then looked at calcium changes. And they found that basically neurons nearby tend to be excited, neurons far away tend to be suppressed. And we sort of see the same thing. A very, very small number of possible interactions actually are significant, maybe 2 to 3%, when you even give 18 action potentials. But cells that are closer tend to be enhanced, and those that are far away tend to be slightly suppressed. These are very small effects. So this is not really a particularly interesting result, but it demonstrates what you could do with the system. What we are particularly interested in doing is actually activating ensembles of neurons. So groups of cells that might have some functional feature tuning in common or some increased correlation in their spontaneous activity. And so again, this is actually just randomly stimulating clusters of cells at a time, same conditions as before. You're imaging 2 planes in the brain, and you're stimulating in those 2 planes. So this is about 5 cells or 5 to 10 cells. And then, as was asked, how much can we push this? So we have a laser that we can get about 4 watts, about 4 microjoules per pulse through the microscope. And here's 3 planes imaged and simultaneously photostimulated. And all the activity you're seeing here is actually generated by the stimulation. And so the arrows flash briefly when we stimulate those cells. And ultimately, we're doing up to 50 cells per shot. We can actually quantify just the delta F over these neurons under different conditions, and that looks something like this. So here's groups, different ensembles, shuffled ensembles of 10 cells, trial by trial, 25 cells, and even 50 cells. You can pretty much reliably activate most of the cells in those clusters. And so this now gives us the ability to begin to activate functionally related groups of cells, or random, as the case as you might desire, as a control, and see what is the functional impact on the brain. Actually, I'm not going to show you that data, because I'm going to summarize where I think we're going with this. So what can we do with the system? So we train in my lab, most of the lab actually studies somatosensation, and we're interested in how animals use their whiskers to discriminate the shape and location of different objects. So we train mice to discriminate different objects with their whiskers, which is their high acuity sense for objects proximal to their head. We measure those patterns of activity using calcium imaging. And then the idea is we can then play those patterns back on a subset of trials in the behavior where we don't actually present the physical stimulus. So on these catch trials, so to speak, we present no physical stimulus, but we play a pattern of activity in the brain. And so if the animal is trained to discriminate, say, a sphere and a cube with its whiskers, which the animals can do this quite well, we can play the pattern for the cube or play the pattern for the sphere back into the brain and see if we can make the animal or fool the animal into experiencing a specific percept. There's definitely a nice literature both of optogenetics and microstimulation of either biasing an animal's perception to a specific percept or really just generating some sort of artificial tingling in their brain if you just stimulate a bunch of cells, and animals will clearly respond to that. What we want is a system that can really fool the animal into experiencing specific percepts, because once we have that ability, we can ask our question about what is the neural code Or what are the features of the neural code for these different objects? So if we imagine there's, again, this raster plot that represents a population code for an object, we want to know what is the syntax, what is the relevant variables in this code that actually correlate, actually causally relate to the percept. And so for instance, if we have that pattern and it's driving the animal to perceive the sphere over the cube, we can begin to shuffle when the cells spike, how much each cell spikes, the synchronization of their spikes, and of course the identity of the cells we're activating, and ask parameter by parameter which of these is most causally related to driving the percept. So this is exactly what we're doing right now. We're training animals to distinguish stimuli with a single whisker, which is convenient because you know exactly where to look in the barrel cortex for that one whisker. And then manipulating what it shouldn't take, barrel has about a few thousand neurons, but it's a very sparse code, so we should be able to gain control of the relevant ensemble in the superficial layers. Okay. So just to conclude, this technology, 3D SHAN, is a new optical approach to synthesize custom neural activity patterns and bidirectionally modulate neural activity. Chrome and IRES GTCR are very potent options for this approach that are optimized for multiphoton holography. We can achieve basically on the order of cellular resolution and millisecond precision to ask these questions about neural coding. And hopefully we can use this technique now, and certainly anyone who wants to can use it in the future to begin to ask their particular questions about neural code in their particular circuit. Okay, so maybe I'll just stop and take some questions before I move on to a separate project. Yeah? That's right. Mm-hmm. That's right. Right, so there are conditions where the imaging does not depolarize the cells. So all opsins have absorbance at a low-pass absorbance. And so that is an issue. What we do to take advantage of that is, first, the opsin is super fast. So if you're raster scanning with a resonant galvo, as we are, the laser flies across the neuron so fast that you get a little blip. So under the conditions that we image, you get maybe under a millivolt. You do get a little millivolt oscillation at the resonant frequency of the scope. That's clearly, what we think, why it doesn't seem to do anything to the cell, at least as we've measured it, is because it's like tiny compared to the background activity. If you zoom in on a cell, the cell will fire a lot because chrome is very strong. In theory, if you could flip-flop and use red and green opposite channels, you'd be better, but there's a lot of reasons why that doesn't work right now. Yeah.
Oh, okay. It doesn't need to be that one. How sure are you that you're generating single spikes with each event? Is it just based on the length of the stimulation? I mean, I'm going to assume, and the data that you've collected from the cell-attached recordings in vivo.
Right. So when we have an electrode, we know. When you're optical, you can't be sure. We can only take our prior based on our data from single spikes. So you can definitely get single spikes if you put a lot of activity in. If GChem could report single spikes, we could experimentally answer your question. That doesn't seem to be the case. We'd like to do voltage imaging. But by and large, most of the experiments we're doing for the moment is not just single spikes. We're actually adding bursts of spikes, which are very detectable. But it's a fair question. OK. So we just finished developing this technology. So I'm going to tell you a story where we use more conventional approaches to ask a question that hopefully is more relevant to the Allen Institute, which is understanding coding and computation of spatial context in the primary visual cortex, in this case of the mouse. And so to give you a little background, contextual modulation is the idea that stimuli placed outside the receptive field of a neuron can modulate the response of that neuron to stimuli in its receptive field. This was discovered many, many years ago, certainly elegantly by Hubel and Wiesel and others, where stimulation like lengthening the bar that would drive a V1 neuron would actually suppress that neuron and not add spikes. So we can call this more generally contextual modulation, and in V1, it's typically observed the phenomena is that of suppression or surround suppression by stimuli outside the classical receptive field. So here's a nice demonstration of this phenomena that we'd like to explain. And the goal of this project is to understand the mechanism of surround suppression. In this case, leveraging the tools that we have in mice. But here in cats, this is in, I think, 2007, in David Furster's lab, they were making whole cell recordings from neurons in a anesthetized cat. And if you give a stimulus that's about the size of the receptive field with the optimal orientation and so forth, the cell fires robustly. But, as had been observed by a number of people, if you make that grading just a little bit bigger so it encompasses the receptive field surround, it's actually much bigger than what's shown here, but let's say like 10 degrees or so, you get a massive suppression of the spiking. You also get a suppression of the memory potential. And so this was an interesting finding that it's extremely robust in the visual cortex, and perhaps the most interesting thing about this phenomena is that if you rotate the surround relative to the center, all that suppression disappears. So it's not the size or the amount of cortex you're activating. There's something special, magical, we'd like to understand, that when the stimulus is large and basically looks like one coherent object, you get massive suppression of the spike rate. So when I was in Massimo's lab, this had not been shown in mice to my knowledge, and it turns out under anesthesia, it's almost non-existent in the cortex or very slight, but you wake the animal up and the suppression in mouse is maybe even stronger than you see in cats. It's really, really robust. Okay, so what is it good for? This is purely conjectural. What other people have speculated, that this mechanism, which is basically widespread throughout many cortical areas, higher cortical areas, as well as somatosensation and so forth, it's good for figure ground segregation. So if you have a grating oriented in front of another grating, you see it as floating in front of that grating. You don't get suppression for the stimulus, but if it's the same grating, you get suppression. So it could help you separate figure from ground. By the same token, it could be important for contour or boundary detection, where you'll get suppression when there is no boundary, but you won't get suppression when there is a boundary. Similarly, visual saliency and pop-out, or more simply, when you have a stimulus that's extremely predictable and redundant, what the cortex does is it basically divides the activity of all the neurons that are responding to that, and you get a much sparser representation for that stimulus, which in theory would be optimal and efficient. But I'm not going to address these ideas so much as the underlying synaptic basis for this phenomena and the idea that it would tell us something critical about the circuit of visual cortex. So what is the mechanism? Well, one idea is that it's a subcortical mechanism, and everything happens in the retina or the thalamus. Now, the fact that it's highly orientation dependent really doesn't seem to agree with that hypothesis, and other features like its time delay. So if it's cortical, it could be a simple mechanism originally proposed was that you get more inhibition. More inhibition would suppress the excitatory cells. Alternatively, you could just have decreased excitation. And a non-mutually exclusive idea is that what actually would be critical is a change in the excitation-emission ratio. So any of these mechanisms could contribute to this critical phenomenon of surround suppression in V1. So in 2007, that same paper from Förster's lab, actually Ken Miller was a theorist who basically they could explain all the phenomena that Förster saw using a network model called the inhibition stabilized network, originally devised by Tzadix. And in this model, the prediction is quite distinct and quite clear, which is that as you give a small stimulus, you generate a certain amount of excitation and inhibition in red here. When you add the surround, you get a very brief increase in inhibition, but then the whole network is suppressed. Both E and I, or excitation emission, go down. And so the net explanation for surround suppression is not just more inhibition with a fixed excitation, but a reduced or a total withdrawal of input to neurons. And in fact, this is exactly what they saw, where the conductance for excitation and inhibition is much smaller when you have a large grating when you have a small grating. And so that afforded a fairly nice description of what might mediate this phenomena. However, it had been long hypothesized, or at least it had been an interesting question, is what are inhibitory neurons in the cortex doing during surround suppression? So as in Massimo's lab, I was recording from the different interneurons because we had crude lines. At the time, we would make loose patch recordings, and we would see that while pyramidal cells and parvalbum cells, and later it was shown VIP neurons, all are very, very strongly surround suppressed, somatostatin cells show much less, and sometimes no surround suppression. So this firing, where it fires more and more to an increasingly large stimulus, suggests a simpler mechanism where you get more GABAergic inhibition. In this case, these are dendritic inhibitory neurons, where you get more inhibition to the dendrites, which would suppress the neurons and generate surround suppression. And indeed, if you inactivate these cells optogenetically, you get a strong reduction in surround suppression. So you get preferential enhancement at the large stimulus sizes. So this actually doesn't directly explain from a synaptic basis what's going on, but it is consistent with a simpler model than the ISN, or the inhibition stabilized network. Okay. So, but I wasn't satisfied because I really wanted to know how we could reconcile the data from David Förster and Ken Miller's paper with that in mouse, where we knew that a specific subclass of interneuron was increasing its firing to explain surround suppression. And so I made intracellular whole cell recordings of excitation and inhibition, so excitation in red, inhibition in blue, and these are two example cells as you increase the size of a grating. And actually, if you look at it, the first thing that's quite obvious for most of these cells, just these two examples, is that excitation is actually highly surround suppressed, just like memory potential, just like spikes. And that is completely consistent with a model of Ken Miller and David Förster. Now, inhibition does show some suppression. It's definitely more modest. But what I wanted to focus on across many cells that was the most consistent finding was that the ratio of the two, excitation and inhibition, would systematically decrease with size. So excitation indeed was suppressed, but it was more suppressed than inhibition. And so in theory, and actually in practice, this could explain surround suppression because you can inject these currents, conductances into a dynamic clamp model, and you would reproduce the suppression of spikes. In fact, in 2015, Miller had another paper where he extended the work of ISN into a superlinear stabilized network, which I won't explain. But one of the major predictions of this model is that the excitation to inhibition ratio should drop with input strength. And input strength can be contrast, and it can be size. And indeed, both contrast and size, you see a reduction in excitation-inhibition ratio. So this is entirely, the data that I collected was entirely in line with the model proposed by Ken Miller. And for the aficionados, this shows the superlinear input-output function of neurons in the mouse cortex that had been shown in cats by Förster years before. So everything did seem to agree. There really wasn't a disagreement, but we had to explain what somatostatin cells are doing. They're firing more and more for larger stimuli. How does this jive? So what I did is when you're making these recordings of excitation, inhibition, I then expressed Hillel-Adesnik in the somatostatin cells and inactivated them. And we knew under these conditions you would get less surround suppression, so more spiking. What could explain that? And so what happened, if you look at here in excitation, when you inactivate somatostatin cells, you actually see a prominent increase in excitation. Perhaps even more paradoxically, you see a very large increase in synaptic inhibition, even though you're suppressing somatostatin cells. And in fact, this is exactly the prediction of the inhibition stabilized network. It's a paradoxical effect that when you suppress interneurons, GABAergic cells, you would get more inhibition. Well, there are admittedly at least two ways you can understand this. But before I explain that, the key idea here is that the reason cells are firing more when you suppress somatostatin cells for larger stimuli is they're getting overall more excitation. They're also getting more inhibition. But because they're getting overall more input, and that input drives the cells fire more when you take out somatostatin cells. Now, the reason you would get more inhibition can either be from the ISN perspective, that if you remove this network suppression, everyone could fire more, and you get more excitation and more drive, in this case, let's say, to parvalbumin cells. An alternative and not exclusive hypothesis is that there's a direct inhibitory circuit from somatostatin cells to parvalbumin cells, and so this would actually disinhibit parvalbumin cells, which target the soma, and give you lots of inhibition. So I'll remain agnostic on how to explain that data, but this is broadly consistent with the inhibition stabilized network model. Okay. So I want to point out in the last few minutes, how am I doing? Okay. That there was a really interesting feature. If you look back at these traces from the 2007 paper from David Förster's lab, if you look at this stimulus here, the large grating, which would suppress the neurons, something really interesting, I thought, which I literally actually recognized like 2 weeks ago, was this amazing subthreshold oscillation that I think hopefully you guys can appreciate that's actually coming at a fairly high frequency. So the low frequency oscillation, by the way, that's due to the grating, the frequency of the grating. But then on top of that frequency of the grating, you do this high frequency oscillation, which doesn't seem to have any relationship to the actual grating. And they did not comment on this, and when I pointed it out to Ken 2 weeks ago, he said, oh, I didn't notice that. So what's going on there? So when we were looking at surround suppression, we were measuring with multi-electrode arrays, spikes and LFPs. We made the stimulus larger and larger and larger, and the thing that was utterly obvious to us on our oscilloscope was a massive synchronization of the local field potential that comes right around 30 hertz. So the low end of what we call the gamma band. So even while the stimulus gets bigger and neurons are firing less, there's a lot fewer spikes in the cells that are representing this part of visual space, the oscillation gets massive. So cells are highly, highly synchronized. So that was funny. But I should say that's been shown in primates and it's been shown in cats. What's really interesting is that if you take that large grating and break it into two gratings, so maybe you perceive them as two objects, these are drifting in this experiment, even though the stimulus is the same size, you get much, much less of this gamma synchronization. So just like you get less surround suppression, you get less surround suppression, more spiking, you actually get less gamma. So the power of this rhythm is inversely proportional to the spiking of neurons, okay? It's definitely not an artifact of increased power, of increased spiking. You're measuring local field potential directly in this experiment that's probably reflective of synaptic inhibition. So most oscillation models depend on inhibitory currents to cells. And we have a model in this paper, but I don't have time to go over it, from Monica Jadi and Terry Stycznowski. But what we knew is inhibition is probably involved. And so if we actually look at the power of gamma, of the inhibitory currents, this is an IPSC, inhibitory postsynaptic current, that power increases with stimulus size, just like the LFP would increase, even while the neurons themselves are being suppressed, and even while total excitation and inhibition are going down, synchronization is going up. So this kind of led us to a somewhat weird hypothesis that we know that as surround suppression increases and inhibition, gamma increases, that all correlates with an increase in the firing of somatostatin cells, and in fact, a decrease of firing of parvalbumin cells, which I haven't directly showed you here. So we made the hypothesis that this rhythm that people, and I think it's the same rhythm that Gray and Singer studied years ago in cats, roughly the same frequency, same features, I'll show you in a second, that this oscillation would in fact be mediated by somatostatin cells, because these are the cells that are firing at the highest rate when the suppression and the gamma oscillation is the strongest. So Julia Viet in the lab, who did these extracellular experiments, expressed Hillel-Adesnik and somatostatin cells. Here's the control, where you turn on a gradient and get a very nice gamma oscillation, a little bump there. You turn on somatostatin cells, and you get this really strong reduction, 10-fold reduction in the total power of a band between like 25 and 35 hertz. You don't see this with parvalbumin and activation, although that's a bit of a tricky experiment. Very consistent across mice. Some of these other things are not super consistent. The most consistent thing is a reduction at 30 hertz. And so what was this good for? Why is there this synchronization? So of course, for those of you familiar with the theory, the idea is that synchronization can phase lock neurons across large distant regions, say of the visual cortex or other cortical areas, and that phase locking per se is a good way of propagating information downstream. So if Julia put two multi-electrode arrays in layer 2-3 of the visual cortex of a mouse, labeled here with a dye, about 600 microns apart, so about 20 or so visual degrees, or a little bit more apart. And she had a grating, she presented a grating that covered the receptive fields of these two electrodes. And so as the stimulus goes on, you see this very strong gamma oscillation. The most important feature of it is that it's super coherent between these two sites. And here's a coherent spectrum that shows that. So very far apart, 600 microns. That's probably longer than most of the axons, except maybe layer 2-3 axon. These neurons are highly synchronized. You can look in the spikes. I'm showing you the LFP, but you can also look at it in the spikes. Okay? But of course, there's also strong surround suppression. So even while most of the spikes have been eliminated by suppression, those that remain are highly synchronous. But if you inactivate somatostatin cells, that synchrony between the two sites goes down substantially. It actually doesn't happen if you have a cross-oriented grading. So what our data suggests is what somatostatin cells are doing is not just generating this oscillation, but they're actually enforcing synchrony across large regions of the mouse's primary visual cortex. So if you have a large oriented edge that crosses the receptive field of two neurons, they're going to be highly synchronized even while many of their spikes are suppressed by somatostatin cells. The remaining spikes that get through are highly synchronized. That could, in theory, be a code for integrating large contours. At least that's the theory. Okay, so to conclude, surround suppression is in fact, and we can now reconcile the data in cats and mice, so anesthetized cats and awake mice. Surround suppression is mediated by withdrawal of excitation and inhibition, and very importantly, it's mediated by a reduced EI ratio. So E is going down more than I, therefore this cell shows more suppression. Sohm cells are actually the neurons that are driving this. They are mediating the inhibition in the inhibition-stabilized network for large stimuli. But just as you are removing spikes, the spikes that you leave behind are highly synchronous with the LFP, but really highly synchronous with each other across large regions of space. And the theory would be, and maybe we can test that now with the right optogenetic approach, that this synchronization is a temporal code for contour integration, which is a contentious idea, but something that maybe we can test. So I want to thank you all for listening. These are the funding sources. And I know that I had a student who came here last week. Maybe some of you saw his stuff. And if you want to have a little more extra data on his project, it might be particularly interesting for those of you doing cell types and on somatostatin cells. But for the moment, I'll just wrap up and take some questions.
The synchronization story, that's mostly observed in anesthetized animals. What? No?
No. That was all awake animals.
Oh, wait. Okay.
Not that there is some oscillations in anesthetized animal. It's not dependent on somatostatin cells. It's kind of a weird oscillation we don't understand. But we're focusing on awake mice.
Do you think about it in terms of efficient coding at all? That on the one hand you have fewer spikes,
Yeah.
right? But because they're synchronized, they're more efficient, right? And so you essentially what do you think about?
Yeah, absolutely. That would be my dominant hypothesis.
Okay, as opposed to the perceptual binding, the contour integration.
I'm not sure those are exclusive of each
Okay.
other.
Yeah.
They could be both. I mean, to put it in a more holistic perspective, I mean, the idea is that when you have this large grading, it's highly predictable. And so if you're coming from the point of view of inference, you don't need many spikes to encode it. And so therefore you have suppression. But you still want it encoded as one contour. And so maybe there you're efficient. You remove redundant information. You make it efficient. But then you use the synchronization as a tool to make sure that there is binding. It's purely hypothetical. Yeah.
Other questions?
Using the first method, so can you synchronize the neurons? So how many or
So how many neurons can we synchronize?
yeah?
So the current limit that we've done per shot, so per frame, is 50.
50.
Squeeze a little more power. There's some issues where if you so again, we illuminate 50 cells for 5 milliseconds. If you try to illuminate, and then you could change that pattern roughly every 4 to 5 milliseconds, pretty reliably. So in theory, at 200 hertz times 50, what is that, 10,000 cells, in theory, if there are that many in your field of view, you could spike with one spike. We typically spike many spikes because we can't even measure one spike with calcium. But when we spike, when a single frame of light comes on, they're going to be fairly synchronized to each other. You could maybe even drive like a little ramp, so you can try to desynchronize them. But in the end, yeah, currently we're limited by that ability to only change the pattern every 5 milliseconds. But in theory, either you could do, it's 10,000 total spikes we could drive in the circuit. It could be 10,000 spikes in 50 neurons, or it could be, actually, that would probably never happen, but it could be 10,000 spikes in 10,000 neurons, but probably more likely somewhere in the middle. Yeah.
Thank you.
So using optogenetics, you create a calcium spike, and it is different from membrane potential. So how do you think about
Calcium spike?
calcium spike and membrane potential?
So, so when you mean calcium spike, you mean dendritically evoked calcium spike. Is that what you're saying?
Yeah.
Well, we don't have any data that occurs, not to say that it doesn't. So we're activating the soma. Typically, when you polarize the soma, you get a sodium spike. And certainly, we measure the sodium spike with a loose patch electrode or an intracellular electrode. That probably backpropagates. You're going to get backpropagating spike. In theory, if you put opsin in the dendrites and depolarize the dendrites a lot, and some people have done this, you can get dendritic spikes. But my guess is that most of what we're seeing is somatically generated, or axon helic generated sodium spikes. Yeah. Although there is some calcium flux through the opsin, if that was your question. That's another issue. We don't actually haven't measured the calcium permeability of chrome yet.
Question up there.
Have you looked closely at the activity of the SOM neurons during these oscillations, and is there any evidence there for maybe different populations of SOM cells?
So the answer to the second question is I don't have any data to say there are different subpopulations in layer 2-3. The SOM cells are synchronized at gamma. They'll fire at a high rate. They could fire sometimes in every gamma cycle, and they fire at a slightly different time than the pyramidal cells and the pyramidal cells, but everyone fires at the trough of the oscillation, just a slightly phase offset. I know there is some data out there about at least 2 or maybe 3 classes of somatostatin cells. I don't know what the transcriptional data says about layer 2-3 in particular. I'm sure Hong Clay knows. And if you're here last week, we have very good data on 2 major subclasses of somatostatin cells in other layers. But at least in layer 2-3, while I'm sure there are subclasses, they're a little more homogeneous than what you'd see in layer 5. But all the gamma oscillation we saw and studied was a layer 2-3 phenomena. It's not very strong in the deep layers, if at all.
June.
How do you think your gamma oscillation compared to the Carding and Chris Moore's PV-induced gamma oscillation?
So I didn't show it in this study. Reviewers asked that, of course. So in that study, they stimulated parvalbumin cells at different frequencies, and they noticed a preferential increase in the LFP power at around 40 hertz. So we reproduced that. You can reproduce that with PV cells, but you can also reproduce it with somatostatin cells at a slightly lower frequency. So both cell types are sufficient to generate this phenomena of resonance, but I would say that the gamma oscillation we observe to visual stimuli, right, in sort of a naturalistic, or at least a mouse that's awake, depends on somatostatin cells. Not to say there aren't many gamma oscillations in the hippocampus, maybe barrel cortex, though we don't see them, that depend on parvalbumin cells. But this particular oscillation, which is a context-dependent, size-dependent oscillation, has all the features that are consistent with somatostatin cells. Yeah.
All right, maybe I'll ask one final question, which is, to what extent do you think that the role of these layer 2-3 somatostatin neurons in surround inhibition will generalize across the cortex to other areas?
Yeah. I think it'll generalize.
All right.
The only thing I could say, because we studied the barrel cortex actually a lot in the lab, but we haven't seen anything like this grating-induced gamma. That being said, you don't have to look at gamma, you just have to look at spike synchronization. In fact, a good complaint for people who don't like gamma is that if you give natural movies, you don't get much gamma. But you still get synchronization, potentially when like a large edge moves over a receptive field. So we haven't done it, but I would speculate that somatostatin cells are going to be generally important across different modalities for synchronization across long distances, because what we have shown, mostly in brain slices, is that they are major targets of the long-range projections of pyramidal cells, and that we know is true also in the barrel cortex, at least. I even did some experiments in like cingulate cortex just for fun in Massimo's lab. And so the basic circuit for generating gamma oscillation through somatostatin cells is widespread. You could see it in many different places by just patching somatostatin cells and driving the pyramidal cells. But whether it's naturally doing gamma oscillations in other areas, I can't comment. Yeah.
All right, great. Let's thank Hillel one more time.